Bismillahirrahmanirrahim. Assalamu alaikum. The topic is Western blotting. Okay. There are three main types of the blotting techniques. The first one is the southern blotting that is used to detect the DNA. Next is northern blotting that is used to detect the RNA, and the western blotting that is used to detect the protein. We are going to study the western blotting because we want to detect the protein of the interest. What basically Western blotting is, the Western blotting, also called protein immunoblot, is a widely used analytical technique used in molecular biology, immunogenetics, and other molecular biology disciplines to detect specific proteins in a sample. In Western blot, the molecule of interest is a protein, and the probe is typically an antibody raised against that particular protein. What basically Western blot is, because we are using we are basically detecting the protein of the interest. What basically protein is, that protein is the antigen or the antibody. We are going to study the antigen antibody interaction or reactions, therefore it is called immunoblot as well. That is used in the molecular biology, immunogenetics, and other molecular biology discipline because for the detection of the protein. Okay, what the molecule of interest is, that is basically the protein, and what the probe is, this probe is DNA or the RNA that is corresponding to the specific gene present in the sample. We have to introduce a probe in that method as well. Okay, basically we are purifying the protein. So this protein purification can be of two types. Either it will be preparative purification or it will be analytical purification. Sometimes we need the protein in a larger quantity. When we need that into larger quantity and we are producing it, that is called the preparative purification. For example, in case of pharmaceutical industries, um, we need the preparative purification. The second one is analytical purification. In this case, we need a relatively small amount of protein. For example, if we are working on research work on a specific protein and we are purifying it, that is in the analytical purification. So basically, we have two different types of purifications, preparative and analytical. The preparative one is to produce large quantity of protein, for example, for industrial processes, and the analytical purification is to produce the protein of interest in a relatively small amount, that is mainly because for the research purposes. So there are six basic steps that are involved in the Western blotting. The first one is the sample preparation. Next is the gel electrophoresis, blotting, blocking, antibody probing, and detection. Let's start with the processes. First of all, SDS page technique is used for the Western blotting purposes. SDS page stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. That means in the electrophoresis process, the gel used is the polyacrylamide gel. The first step is the sample preparation. In the sample preparation, as the name indicates, the sample is prepared for the detection of the protein. We take the, for example, if we have to disrupt the cell to like take out the protein to take out the protein from that cell. For that, we use different types of physical or chemical methods are used for the destruction of the cells, so that DNA and RNA and all the other constituents get destroyed and only the protein is left. After the sample is prepared, that means the DNA, the protein is isolated from the sample. Gel electrophoresis is used. The sample is then run on the gel electrophoresis using polyacrylamide gel. The proteins of samples are separated according to the size on the gel in the gel electrophoresis. What basically happens in this gel electrophoresis is we prepare the gel, that is a sodium, that is a polyacrylamide gel. These holes are made, these wells are made with the help of the foam. Buffer solutions are poured and the samples are poured in these wells or introduced in these wells. We have a dye that is used for the visualization of the samples. The samples, when it is connected with the positive and the negative electrode, the samples like cover some distance. The bigger samples will cover smaller distance, whereas the lighter samples will cover the larger distance. Based on the size of the distance traveled, we differentiate the proteins of interest on the basis of the size. Okay. Gel electrophoresis, loading samples and running the gel. Samples are loaded into separate wells. A protein marker is also loaded, run at 200 volts for 30 to 40 minutes, and bromophenol blue dye is used to visualize the samples. Okay. What basically a protein marker is? A protein marker is that protein sample whose size is known to us that is used for the estimation of the size of the other proteins. Now, this is the pipette that is used to introduce the samples into this well. The, in the process of electrophoresis or in the molecular biology more specifically, pipetting skills are very important. The more like expertise in the pipetting skills are, the more are your results accurate. So these wells are loaded with a sample along with a protein marker, and they are run at 200 volts for 30 to 40 minutes, and bromophenol blue dye is used to visualize a sample. Bromophenol blue is a solution that contains a slight negative charge. The next step is blotting. Now, this blotting step is very important in the all the blotting techniques since the process is Western blotting. Now, the proteins in the gel are then transferred onto a membrane made of nitrocellulose by applying current. We smoothly take out the gel in the gel electrophoresis chamber. We pour it, and a nitrocellulose membrane is used for the transference of this gel samples. Why is nitrocellulose membrane used? Because it has the high binding capacity. Therefore, the sample on the gel are very smoothly transferred onto the nitrocellulose membrane. The next step is blocking. Blocking is a very important step in the immunodetection phase of Western blot because it prevents non-specific binding of antibody to the blotting membrane. Commonly used blocking solutions used non-fat dried milk in a solution of phosphate buffered saline. Okay. Now, blocking is important. Why it is important? Because it prevents the binding of the non-specific antibodies onto the blotting membrane. What is non-specific binding? That it will not bind, it does not have epitope of the specific antibody. Therefore, blocking is important so that the rest of the antibodies should not bind to the proteins. Next is the incubation. Incubation occurs by diluting an antibody into a solution which will keep the pH neutral. The majority of the time, the solution is non-fat dry milk. Okay, we just primary antibody is incubated into the membrane, and the primary antibody recognizes only the protein of interest and will not bind any of other proteins on the membrane. The protein of interest is present in the sample. Primary antibody is added to the sample, that is added into a solution. That solution is non-fat dry milk. When it the function of the primary antibody is that it will only recognize the protein of interest and it will not bind to any other proteins present in the sample. Next is the antibody probing. The blot will be incubated in a dilute solution of antibody, usually for a few hours at room temperature overnight at 4 degrees centigrade. The solution that has been diluted with the antibody, it will then be incubated for overnight at 4 degrees centigrade or a few hours at the room temperature. The last step is the detection. The detection is done by using the X-ray film or CCD camera for light capture, and the production of purple black bands directly on the surface of the blot will confirm the presence of the protein of the interest. So this is the simplest Western blotting technique, in which first the sample is prepared and then gel electrophoresis is done using polyacrylamide gel. Blotting is done on the nitrocellulose membrane. Blocking is done for the prevention of non-specific antibody. Antibody probing is then done, and at the end X-ray film or CCD camera is used for the detection purpose, and at last the result is obtained as I guess here was the result somewhere. Okay, and at the end, the result is obtained as purple or black bands that confirm the presence of the protein of the interest. If still there remains any query, you can contact me in the comment section. Thank you so much.